Welcome, everyone, and thank you for joining us for our webinar titled A New Frontier of Precision Medicine Using PET for Image-Guided Neurointerventions. This is Liam Sanyo from Inside Scientific, and I'm very pleased to be your host for today's event. Our session is sponsored by Syntica Instrumentation and will feature Dr. Miroslav Janowski, Dr. Piotr Walczak, and Dr. Wojciech Lesniak, who will discuss how PET-CT can be used to study the targeted delivery of therapeutic agents to the brain for image-guided neurointerventions. Excellent. All right, so without any further ado, I'm very pleased to introduce our first presenter, Dr. Miroslav Janowski, or Mirek. Mirek, thanks so much for joining us today.
First of all, I would like to thank Syntica and Sedical for organizing this event in these absolutely unprecedented times of COVID-19 pandemic. And while we put the PET on the front page, as we've been recently most excited about it, it would be difficult to catch its importance without a story, with a background story and our journey, how we've got there. On the preclinical side and at the end, we'll touch a little bit where we are on the clinical side. Central nervous diseases, including neurological, oncological, and psychiatric conditions, altogether cause the biggest chunk of healthcare expenses. Thus, the delivery agents to the brain is a highly compelling topic. Systemic delivery, peros or intravenously, is always our first thing to think due to easy administration, but because of blood-brain barrier shielding brain from the body, we typically need high doses of therapeutic agents, which are typically characterized by high toxicity. Intracerebral or stereotactic delivery is on the other side of this extreme. You are certainly beyond the blood-brain barrier, but you need drill the skull and puncture the brain. In addition, the spatial biodistribution of agent is frequently limited, and it's quite difficult to repeat the procedure if needed. Intrathecal route is something between, and it has certainly better ratio of positives to negatives, and it's attractive for whole brain delivery, like antisense oligonucleotide in Huntington disease, for example. It's now in the clinical trial or now at routine treatment. But still, drug targeting to specific areas far beyond the capacity of this route. Intra-arterial route seems to have even more advantages, such as focal delivery, but at the same time to large territories if needed. So there is a lot of flexibility. Thus, it's our favorite neurointervention. It's relatively minimally invasive, and it can be relatively easily repeated, even tens of times. However, I do hope to convince you that it needs really advanced imaging to be fully exploited. Obviously, to deliver drug directly to vessels feeding the lesion is not a new concept, and actually it has been for the first time attempted nearly 70 years ago. Clinicians were brave enough to administer even such toxic agent as a nitrogen mustard, as obviously at that time there was no better choice. That approach couldn't be successful though. But this route was so intuitive that it was reattempted by other groups with newer and less toxic agents such as methotrexate, mitomycin, vincristine over the next nearly 30 years. Until the breakthrough paper has shown that intra-arterial delivery of osmotic agents such as a simple mannitol is capable safely and for a short time, temporarily, open blood-brain barrier, and by that way to skyrocket brain penetration by therapeutic agents. This discovery has been accomplished by Dr. Newell, who is here the first author, who currently works at Oregon Health and Science University. And he was so kind that accepted our invitation to be a panelist at our SIGN conference last year in Baltimore. I will tell you a little bit more about the conference later on during the webinar, but notably it took another 20 years to perform and publish a multi-center clinical trial using this method. More than or almost 2,500 procedures were performed with only one procedure-related death. So it's pretty much safe procedure. Patients with a very rare disease like primary CNS lymphoma responded excellent, and it's a method of choice for treatment in many clinical centers worldwide, but it's a rare disease. The results with the most frequent and nearly most malignant brain cancer were also encouraging. However, the same year, published analysis revealed the huge variability in BB opening across patients, which added the high variability to the treatment process. This could be a reason that Medicare killed the procedure in the memo published in 2007, unfortunately. But even this decision didn't quell the enthusiasm of scientists and clinicians, especially as technological advances with new, more flexible catheter and safer drugs such as biologics, including antibodies, caused a new opening, especially with possibility for performing a procedure more precisely with an ultimate goal to eradicate variability. Here actually the group from New York has confirmed that super selective delivery of drug to the brain is feasible and safe. So using X-ray to guide the procedure, it was necessary just to guess where exactly the drug is given. Next, my colleagues will present our preclinical journey, how to visualize where the drug is given and how much of it. And at the end, I will come back to show that it is now possible to go beyond the guess and visualize the brain territory exactly to which drug is given intra-arterially.
Hello, everyone. So I will take it from here. Mirek gave a really nice set the stage and showed that the advantages of the intra-arterial route to deliver therapeutics to the brain, and spoke a little bit about the challenges that this one is facing when using this approach. So now I will show you how we are trying to address the challenges and make it more reliable and reproducible approach. And I will start with some more examples why we are so excited about intra-arterial delivery and why we think this is the way to go with majority of applications with drug delivery to the brain. So on these images here, I'm showing you two studies. On the left, there is a preclinical work published by Ushida in 2012, and it is showing a mouse brain transplanted with stem cells, transplanted intracerebrally with stem cells. And you can see that the engraftment of these cells is outstanding. The therapeutic effects that were observed were great. And based on that, there was a clinical trial performed in kids suffering from myelin disease. And here you can see that in this brain, these arrows pointing to the two hypointense regions show presumable positive effects, myelination. And I'm just showing you this to demonstrate how limited is the distribution of the cells or this would be also for other therapeutic agents when we jump from small animals to patients. And that is something that we have to be really aware. The size disparity, there is between mouse and human, there is 1,000 times difference, and we need to use adequate technique for delivery. We cannot rely purely on small animal studies because the size is will cause some significant issues. So here, when you see on the left this vast vascular network, it gives excellent opportunities to target. And as Mirek said, there are very good techniques, and the vascular techniques, even more quite distal arteries can be selected for targeting very specific regions. And on the cartoon on the right, it schematically shows what we want to get. We are putting the catheter into the selected artery, and we assume that when we inject therapeutic agent, its distribution will be homogeneous downstream from that injection site. Unfortunately, this is not the case. It's more challenging than that, and we learned from decades of clinical studies. This graph was shown by Mirek. showing huge variability in drug accumulation and blood-brain barrier opening after intra-arterial injection. And I started working on this intra-arterial approach many years ago. In 2008, we had a paper showing these challenges in small animals, in rats, with injection of iron oxide labeled cells. These hypointense regions correspond to intra-arterial injected cells. And from this busy slide, I want to just convey the message that what we observed was huge variability. There was a very high engraftment in some animals, some moderate engraftment in other, but about 50% of animals, there was no accumulation of cells, no cells found in the brain at all. So over the last 10 and more years, we are trying to address this, to understand and to improve reproducibility. So here I'm going to show you the multimodality imaging platform that we established and we are developing further. And it is now we use various animal models, but I'm going to focus here on mice. So in mice, and since we are going to go across different imaging modalities between MRI to photon microscopy, PET, we want to use tools that are compatible across. And here is an example of cranial window that we adapted, which is all non-metal components that we can go between MRI and two-photon microscopy. We then placed catheter into internal carotid artery. So in mice, obviously, you cannot go very distal into the brain, which is possible in humans. All the extracerebral branches are occluded for safe infusion into the brain. Then we place animal inside of the high field MRI scanner. And what is important that we have infusion pump just next to the gantry, so we can perform infusions of agents while acquiring MRI scans. And then the next steps include intravital microscopy and PET imaging that Wojtek will talk about later on. So here, I just want to show you an example in a very straightforward way how we visualize transcatheter perfusion. So the mouse is in the scanner. We have this infusion pump, and when we do fast T2 star imaging with a very fast temporal resolution of 2 seconds per volume, we can see oh, well, unfortunately, this movie doesn't display properly. So you will have to believe me that you see hypointensities. This part of the brain turns dark. And these hypointensities can then be segmented. And here on a color-coded image overlaid on the T2, we can visualize what part of the brain is perfused. And what is interesting that depending on the infusion rate, we have situations that even though we inject into carotid, there are situations that brain is not effectively perfused. When we increase that rate, we can effectively perfuse the brain. And at that point, we know that we can start injecting therapeutic agents. And I will talk about this a little bit more on the subsequent slides. One important issue with intra-arterial injection is safety. There are several situations when this can be harmful, and one is speed of infusion. Excessive speed can be damaging, and in this case, I'm showing you 200 microliter per minute speed in mouse is causing hyperintensity, 2 hyperintensities on MRI, which indicate damage. When we look in histopathology, there is astrocytosis and microglial activation. So this is something that we want to avoid, and we can when we adjust that infusion speed to the safe level, which we established as 150 microliters per minute. And now, example that I want to show you how we can see and verify this, some reasons for variability that is observed in animals and also clinically. In this case, on the left, you can see vascular anatomy, and in this particular case we cannulated carotid artery, common carotid artery, but there is a hard to get branch pterygopalatine that in this case we left open, assuming that 50% of the cells will go to the brain, 50% will go outside. When we did MRI and we looked at distribution of iron oxide labeled cells, we found that hypointensities are found, all of them are found outside of the brain in the supply of the PPA. So that is showing you the challenge, the complexity of cerebral vasculature. The other arteries supplying the brain cause resistance that will have to be overcome for the cells to get in. But in another experiment, when we close this extracerebral branch, we can have effective and proper targeting into the brain. So not having imaging, we would assume the cells are or other therapeutics are getting to the brain, which is not always the case. Here, another example, even if you have effective perfusion through the brain, you want to see if the cells or other therapeutics are engaging and accumulating in the brain. Here on the left, you see the experiment with real-time MRI imaging that is showing that the cells were injected, but they passed through, they did not stop in the brain, while here different type of cells, they do engage with cerebral endothelium, accumulate in the brain, which you can see on these graphs when we quantify signal intensity. The signal remains low here and then recovers after some reduction. So it's very important to know what is happening over time as you optimize your parameters, your experiment to have effective accumulation in the brain. So now injecting cells intra-arterially, we want to make sure that the cells extravasate and integrate with the brain. So this is a T2 MRI before injection. After injection, you see these characteristic hypointensities of iron oxide labeled cells. In histopathology, you can see red labeled cells that appear to be outside of the green blood vessels. On a high magnification, you can appreciate it even better that many of the cells appear to be outside of the brain, but ultimate proof we performed using two-photon microscopy, intravital microscopy, where in the live animals we observed animals over 30 minutes, 50 minutes, up to 2 hours. And over this time, if we look on these cells, green labeled intra-arterial infused cells marked with arrows, and if you can follow them over time, you can see that they transition outside of the vasculature. And even more beautiful proof that they indeed extravasated and integrated. It's here at 2 weeks, this GFP positive cells beautifully integrated with the host brain. So now, very quickly switching gears and demonstrating how we can use and how important it is to use imaging for controlled opening of the blood-brain barrier. Blood-brain barrier is there for a reason, but it is an obstacle to drug accumulation. Majority of drugs larger than this size and hydrophilic, they do not extravasate well after systemic injection. So what is needed, and what Mirek already mentioned, is a technique to open this tight junction, this blood-brain barrier. So injection of hyperosmotic mannitol is causing that transient opening that lasts about half an hour up to 1 hour. And then in an experimental outline here, what we are doing, we have a mouse, we cannulate the carotid artery, we place it inside of MRI for image guided opening. We can pre-inject a contrast agent to visualize transcatheter perfusion territory and adjust that. Then we inject mannitol to open the barrier and verify opening with gadolinium. Here are some results. So this pre-injection, where we probe which part of the brain is perfused, this is here. You can see this hypointensity that did not display in the previous slide. Then when we inject mannitol and do T1 after gadolinium, you can see quite good correlation. And when we do histology, with a gold standard Evans blue or fluorescence for IgGs, we can see indeed effective opening of the barrier. Safety is important, so when we check several days after opening, the barrier reseals, there are no damages if injection is done properly, and there is no inflammation in histology. So I think I have now a couple of more slides to show you how we use imaging, MRI, to optimize the procedure that we can effectively and safely do two-photon microscopy, intravital microscopy. As you know, this intravital microscopy requires placement of cranial window, and we can only see very shallow, a couple hundred microns into the cortex. So it is essential that we can open the barrier, we can get effective perfusion through the catheter in the cortex. What is quite unfortunate and was surprising for us is that when we inject into carotid artery, in almost all animals, we do not have perfusion through the cortex. It is apparently perfused by the contralateral hemisphere. Majority animals, we get only deep structures that are perfused. This is caused by this complexity of the vasculature and non-terminal vessels in the brain. But we found a method to force opening of that perfusion in the cortex, and we do it through transient closure of a contralateral carotid artery. While we infuse agents into the ipsilateral, for that couple of seconds, 30 seconds, 40 seconds, we occlude the contralateral, and you can see on this dynamic MRI that after opening, the cortex is, after closing the contralateral, the cortex is perfused, what you can see on this time course and on the graph. See, so there is no perfusion in the cortex, we close the contralateral, and we can see effective perfusion, the hypointensity in the cortex. So then we can do two-photon microscopy, high resolution, we can see this beautiful vascular network when we inject FITC-labeled antibody after mannitol. You can see this spilling of the agent outside of the vessels. So when quantified here, you can see when, to what extent these agents extravasate. The red is small molecule rhodamine, the green is antibody. You can see the small molecule extravasates earlier, which is quite intuitive. In histopathology, when we do not open blood-brain barrier, when we have it intact, there is still some advantage of intra-arterial injection, but the agent is inside of the blood vessels, shown here on the high magnification. But when we open osmotically, there is a lot of transition and extravasation of antibody into parenchyma. So to summarize, MRI is excellent and very important to visualize this process of infusion and adjusting cerebral perfusion territory. It cannot be done with X-ray because X-ray angiography is not sensitive enough for parenchymal. MRI is great for that. BBB also we can monitor with that. Intravital microscopy is also outstanding tool that helps to visualize the cellular processes. But these two methods, MRI and intravital microscopy, are not really quantitative. And here where PET is coming in, and Wojtek will talk about this now.
Pleasure to be a part of this webinar, and to start with, I'd like to thank Mirek and Piotr for excellent staging up of my part of the presentation and emphasizing the importance of application of PET. Although they obtain a lot of interesting, important results based on MR, it cannot be used to estimate the delivery of the drugs themselves to the brain, because it is very difficult to radio-labeled drugs to be visualized by MR. There are attempts to radio-labeled antibodies, and they used super paramagnetic iron oxide nanoparticles, which are very large, around 100 nanometers in size, between 150 actually nanometers. And the conjugation of such a large construct with antibodies definitely affects the pharmacokinetics and pharmacodynamics of the antibodies. And importantly, to be detected by MR, you have to reach a range of millimolar concentration, which is very difficult to achieve in vivo. On the contrary, the radiolabeling of antibodies and other small molecules and other constructs, cells and other nanoparticles, involve very little modification. Like shown here, antibodies can be radiolabeled via conjugation of very small chelator and subsequent chelation of zirconium or copper, depending on the needs. And this little modification definitely does not affect pharmacokinetics as well as the pharmacodynamics of antibodies. And PET is highly sensitive. It can go down up to picomole concentration of the radiotracer to be detected. And importantly, it is quantitative as opposed to MRI. So how the positron emission tomography works? There are a number of different radioisotopes with different half-life times, which can be either covalently attached to the biologically active molecules, or we have radiometals which can be chelated to the drugs and imaging agents. Then when the radiotracer is ready, they are injected systematically into the vein, into the patients, and then they decay via emission of the proton, which travels very short distance in the body, collides with the electron, and during this annihilation process, two photons are produced, which are sent in two different directions, which are picked up by the detectors, and the signal is reconstructed to get a 3D distribution of the radioactivity in the body, which can provide a lot of important information depending on the use of the radiotracer. And to start our collaboration with Mirek and Piotr, we selected bevacizumab because it was FDA approved for treatment of recurrent glioblastoma, and it was also used in intra-arterial delivery in patients for treatment of recurrent glioblastoma, and showed excellent therapeutic efficacy and much lower toxic side effects. So how the radiolabeling of bevacizumab involves a conjugation of DFO, and we used MALDI to quantify how many DFO molecules were conjugated with the antibody, and it indicated that around 3 molecules, on average 3 molecules of the chelator was conjugated to bevacizumab. Then using ELISA assay, we have demonstrated that intact bevacizumab and the conjugate bind to its target to the same extent, and then we radiolabel this with this construct with zirconium, which involves formation of thermodynamically stable complex, octahedral complex, shown here. And the cis-exclusion chromatography shows that the radiolabeling was done properly because of the coelution of intact non-radioactive bevacizumab with the radiotracer. And on the next slide, you can see the experimental setup. First, mice underwent the surgery involving the placement of the catheter into the internal carotid artery, shown here in black. And then on the other side, the catheter was connected to the infusion pump. Next, mice were transferred to our CIDICAL Super Argus small animal PET imaging, PET CT imaging system, and we have investigated 3 different groups involving intra-arterial infusion with intact BBB. In another group involved the osmotic blood-brain barrier opening with a mannitol followed by intra-arterial infusion of the radiotracer. And in the last group, we opened blood-brain barrier with mannitol, followed by the IV infusion of the radiotracer. In this case, mice had two catheters, one placed in ICA and the other one in tail vein. So upon initiation of the infusion of the radiotracers, we started dynamic scans, lasted half an hour, composed of 60 30-second frames to evaluate early delivery of the radiotracers into the brain and the initial retention in the brain. And we have evaluated three different macromolecules: antibodies, nanobodies, and polyamidoamine dendrimers. On this slide, you can see the results which we obtained for bevacizumab within first half an hour. As you can see, the highest uptake was observed in the group which involved opening of the blood-brain barrier and IV and IA infusion of the radiotracer, which reached around 25% of injected dose per cc within 5 minutes of infusion and persisted at the same level for the next half an hour. In another group involving IA infusion of the radiotracer with intact BBB, we achieved around 10% ID per cc. And in the last group involving opening blood-brain barrier and IV infusion of radiotracer, we just detected the background of the radioactivity in ipsilateral hemisphere. To visualize this uptake, I sum up those 60 frames, and you can see a nice uptake in the brain in a ipsilateral hemisphere of bevacizumab. And moving forward, we have used PMOT to evaluate the distribution of the radioactivity in different brain parts. So as you can see on this graph, the highest uptake was observed in striatum, hippocampus, and amygdala. And the same follows for this group involving IA infusion with intact BBB. So, and then here you can see the whole body volume rendered PET-CT imaging, confirming the results of very high uptake of the bevacizumab at 1 hour post-injection. And 24 hours post-injection, we still can detect the bevacizumab in the brain. And importantly, the decrease of the concentration of bevacizumab in the brain was only by around 3% in this group. And then we have ex vivo biodistribution, which confirms the results shown in previous slides based on PET-CT imaging. As you can see, the highest uptake was of the radioactivity was in ipsilateral hemisphere and compared to contralateral. And due to the long circulation time, we have high concentration of bevacizumab in circulation still at 24 hours post-injection. Then, those results were published last year as a featured paper in Journal of Nuclear Medicine, which actually was published together with the invited perspectives written by our peers from Netherlands, who emphasized the importance of the application of PET in those kind of procedures, and especially now where we have 4 clinical trials involving IA deliveries of the drugs in the patients with different brain tumors, and the 3 of those clinical trials involve the administration of antibodies, which can be easily radiolabeled with zirconium, as I have shown before. And encouraged by those results, which I just showed, we moved to nanobodies. Nanobodies are much smaller than antibodies, and that's why they can pass through the biological barriers easier than antibodies, and we use the same approach to radiolabel the antibody as we used for bevacizumab. And then indeed, we have detected humongous uptake of the nanobody in the brain, shown here. Whole hemisphere was lighting up, and you can see the much smaller uptake of the radioactivity in kidneys. And then IA delivery with intact BBB, we could also see nice uptake reaching around 30% of ID per cc, but you can also see higher uptake in kidneys. Interestingly, opening blood-brain barrier followed by IV injection resulted in a higher activity uptake in kidneys and lungs 1 hour post-injection. And here you can see the whole body volume rendered images showing a nice uptake of the antibody in this group, a nanobody in this group, and then 24 hours post-injection, you can see decrease of the radioactivity in each lateral hemisphere, and quantification indicated that around 50% of the nanobody was cleared from the brain, indicating that the nanobody not only goes to the brain easier compared to bevacizumab whole antibody, but also it's cleared from the brain faster than bevacizumab. And now I will switch gears and we'll talk a little bit about polyamidoamine dendrimers, which are one of the most versatile constructs to prepare nanoparticles for different biological applications. Their synthesis in base is on two repetitive reactions. First, Michael addition of methyl acrylate to ethyloδιamine, followed by amidation of resulting ester. This leads to the formation of generation 0 dendrimer, and then repetition of those two reactions leads to the formation of next generations, generation 1, generation 2. The size of generation 1 is around 1 nanometers, which has 8 terminal means. Upon formation of next generations, the size of dendrimers increases by 1 nanometer, and the number of primary amines is doubled. Starting from generation 4, we have 64 primary amines on the surface, and then globular macromolecules. And due to the large number of primary amines, we can conjugate a lot of different functional groups to the same dendrimer. Targeting agents, therapeutic agents, contrast agents, you name it, we can conjugate. And the surface of those dendrimers, it can be easily modified using acylation, carboxylation, hydroxylation, or pegylation. And importantly, the interior of dendrimers is hydrophobic, which can be used for encapsulation of hydrophobic drugs or formation of the metallic nanocomposites for different biomedical applications, I would say. And then we have selected, for our studies, we have selected generation 4 dendrimer with theoretically 64 primary amines on their surface, and then we conjugated 3 molecules of DFO with it, and then hydroxylated the surface of dendrimers via reaction of remaining primary amines with glycidol, and this construct was radiolabeled with zirconium again. But keep in mind, those are around 4 or 5 nanometers in size nanoparticles, very hydrophilic, with uniform surface properties. We have only hydroxyl groups on the surface, mostly. And then, as you can see here, on the contrary to the antibodies and nanobodies, dendrimer, regardless the status of the BBB, went to the brain at very small extent, around 4.5% of injected dose per cc, and it showed a very fast, steep clearance. Within half an hour, everything was gone from the brain. And here I just sum up the frames from 5 to 10 minutes just to show some uptake of dendrimers in those two groups. However, 1 hour post-injection, even bumping up the scale, I couldn't see any dendrimer in the brain. So all radioactivity could be detected in kidneys, bladder, and liver and spleen. And whole body volume render imaging confirmed those results. Then we have ex vivo biodistribution showing background level of the radioactivity in the brain and most activity in kidneys and liver. So this is the slide which summarizes our results obtained based on PET. And although we use exactly the same method for the delivery of those three different molecules, we observed vastly different delivery and retention in the brain, which is emphasizing the importance of the application of PET to understand how much of the drugs or delivery vehicles as dendrimers could be delivered to the brain. And I'd like to also point out that the results obtained for hydroxy-terminated dendrimers non-targeted is quite encouraging, because we can prepare targeted dendrimers using targeting agents for different receptors overexpressed in the brain or brain tumors and deliver them there. What I mean by that, shows this slide. We have recently published a paper in which we are describing the synthesis of the same dendrimer based generation far, but PSMA targeted. And for targeting, we used lysine, glutamate, and urea-based targeting agent, and the PSMA is overexpressed in most of the prostate cancers and on endothelial cells in tumor microvasculature of different tumor types. Then we conjugated rhodamine for optical imaging, then DOTA for the radio labeling with copper-64. And as you can see here, mice having two tumors, we have much higher uptake of the nanoparticles in targeted tumors versus non-targeted tumors. And the same was confirmed by optical imaging and the microscopy, showing here in red the internalization of the nanoparticles into the tumor cells. In another set of experiments, we have prepared the PSMA-based dendrimers for dual imaging of PSMA expression by optical imaging, shown here, which is indicating highly selective uptake in targeted tumor versus other organs. And then here the slices of the tumor shows that the dendrimer was distributed throughout entire volume of the tumor. Actually, to be honest, I have never seen nanoparticles which can so easily go through entire volume of the tumor. And the same nanoparticles could be detected by a quickly emerging novel photoacoustic imaging, shown here in red. So what I'm trying to emphasize here, that we can take the advantage of highly specific focal and efficient IA delivery of the agents, dendrimers, to the brain, and then active targeting, which is very specific. So with that, I'd like to thank you for your attention and welcome Mirek to continue our presentation.
So now you see it is a promised volume rendering in the first patient treated with neuroendovascular procedure performed under real-time MRI guidance. So when I was finishing, I was showing just X-ray where you have vessels and actually you don't know where exactly it's going, what you are injecting. So here you can finally see it, and this is like the following what Piotr was showing. So here we see with the with blue color segmented brain cancer, with yellow the territory to which drug was given, and the overlap of infusion area and brain volume, which were infusion, was demarcated with the purple color. And then here we see the difference in tumor coverage depending on the infusion speed, which was visible in real time during the procedure. So we see here the differences between the faster infusion and slower infusion. That's why we can finally, during the procedure, manipulate and to see in real time what we are doing. If anyone is more interested in this case report, I recommend to look into a full story published in top journal in the field of neurointerventional surgery. Apparently, the editors were quite excited with our paper, as the image that you have seen before has been put as a cover image, which is actually very rare for case reports. So that's kind of unusual. And obviously, the next step after MRI, we would like to go with a PET with the same concept. Then now already to briefly summarize, after our webinar, I believe that no one will doubt that the intra-arterial route of delivery of therapeutic agents to the brain is becoming increasingly attractive, and advanced imaging is essential to perform the procedure precisely, and by that way to eradicate variability. It was all possible with new advances, which emphasize the advantage of endovascular approach to the drug delivery to the brain. However, there are still things to do. We need to design MRI visible microcatheters for endovascular neurointerventions. We need to engineer cells and molecules to better benefit from intra-arterial delivery, and application actually of PET MRI in interventional suits to benefit from advantages of high spatial resolution of MRI and high sensitivity of PET during the same procedure to allow for unprecedented precision. And here, coming to acknowledgements, obviously our work wouldn't be possible without many people and generous funding from actually all over the world. So I believe here the whole world needs to put together the efforts, the same as now with the COVID-19. The one country is just not possible to make a breakthrough. And really, since the brain is a really different world, we have launched a new initiative of Society for Image-Guided Neurointerventions, SIGN, registered in state of Maryland in the USA, which organized already 2 conferences in 2018 in Warsaw and Poland, and in 2019 in Baltimore, USA. And let me here share a few pictures from this event, which I think was quite successful. And we have planned another SIGN in 2020, this year in Warwick, UK. But due to pandemic, we had to push it till July 2021. So stay tuned, please. As we expect after a year of break, it will be another successful event. I've seen today that the Nukin, from the Trump team, has announced that probably it will be difficult to go outside of the country until the end of this year. And we are also closely working and organize previous event, and we'll organize the next SIGN conference with the Children Brain Tumor Drug Delivery Consortium based on the University of Nottingham in UK. And please get to the website and register with us here. Thank you very much.
All right, thanks so much, Mirek, for the fantastic presentation. And so with that, I'd like to invite the other presenters back onto the audio line, and we can get started. So our first question today comes from Oscar, who asked whether you can measure 2 isotopes at the same time. And Wojtek, I think you were, this question came in during your presentation.
Yes, actually you can measure 2 isotopes, but you need to use PET or SPECT, to one radioisotope. So you need to have 2 different scanners, and you cannot measure simultaneously 2 different radioisotopes. So,
okay.
I mean, we were thinking also of using like a very short half-lifetime rubidium to monitor the blood-brain barrier opening, which decays within a second, and then to follow with the therapeutics. But it is impossible to be honest.
Can I jump on that? So actually, there is now new methods that are capable to detect true radioisotopes by PET, but still it needs to be one that produces gamma rays like a zirconium, and then another one that is not producing like FDG. So if you have the same like a carbon and FDG, you cannot, but you could zirconium and for example FDG or carbon. So that's a very new advances in this field, and I think that will be more and more application for that.
Interesting. All right, thanks so much, Wojtek. And Mirek, and yeah, so I'm hearing that you can use two isotopes at the same time. That's interesting. Another question here that was asked soon after Piotr's presentation, so maybe I'll direct this one to you. But how do you measure immunoreactivity after modification?
So I'm trying to understand what is meant here. Modification is, I would guess that this is after labeling for MRI, labeling for PET. So there was a lot of studies looking into impact of labeling stem cells with iron oxide nanoparticles, and while there are effects, including changes in migration of the cells, the differentiation in some of the lineages, these differences seem to be transient. And overall, it is considered that labeling cells with iron oxide nanoparticles is pretty safe. Then labeling molecules, macromolecules for imaging, and this would be particularly for PET imaging. I think the most the biggest concern is that how that would impact pharmacokinetics and pharmacodynamics. So loss of activity is a big concern, but obviously also the immunoreactivity. We didn't do much of these studies. Immunoreactivity studies. We are more worried at this stage on activity and if the drug still can engage with its target.
Excellent. Great answer. Thanks so much, Piotr. And I think in the interest of time, we'll try to squeeze one more question in here from Tamara, who has asked, how does FUS, or functional ultrasound of the blood-brain barrier opening, compare to mannitol or artery ligation? Do any of you have experience with this?
I could try answering this. So focus ultrasound is definitely very interesting technique, gaining interest recently, and I'm sure these various techniques will find their niches. We already identified some major differences between focus ultrasound and osmotic technique. One of them is the duration of the opening. With mannitol, it's very short. It's 20 minutes, maybe 40 minutes. With focus ultrasound, it takes days. It's also difficult still to control the energy, and still there are some concerns about damage after focus ultrasound, while there is a lot of experience, including clinical experience with mannitol, that it is completely safe.
Excellent. All right. Thanks so much. And I think we'll have made that the end of the Q&A session. But thanks so much, Mirek, Wojtek, and Piotr, for your excellent contributions, both in the presentations as well as the Q&A session.
My pleasure.
Thank you very much.
Thank you very much.
Yeah, my pleasure. So thank you, everyone, for taking time out of your day to attend our webinar. And we hope that you found the information presented both educational and applicable to your research. And of course, a very big thank you to our sponsor, Synteca Instrumentation. So in closing, thank you again for taking part in this Inside Scientific webinar, and we look forward to having you with us again soon.